Yes, guys, good morning. Today we have the second lecture from cell cycle and cell division. Hope you all are perfectly fine and ready for the class. Yes, we are going to start now. Hope the screen is perfect. Yes. Okay, then we are moving forward to today's lecture meiosis, part 2 of cell cycle and cell division. Before that, if anyone new is watching this session, kindly join our Telegram group at Bypoint New, where we conduct the regular discussion, polls, daily questions, updates on classes, OMR exams. And guys, just join our Telegram group to know more about the crash course for Neet 2021, which we are conducting now along with this session too. Guys, do not forget to hit the like button. Do share it with your friends. Do subscribe our YouTube channel by Stream the World of Bio. And do not forget to leave a comment in the comment section. Okay, then, guys, we are going to start today's session on meiosis. Last class, we have learned about mitosis, which is known as the equational division. And now we are going to start with meiosis, which is known as the reductional division. Meiosis or reductional division. It reduces the chromosomal number. That is, the normal chromosomal number is 23 pairs, right? 23 pairs is the normal chromosome number. It get reduced to 23 in number. 23 pairs, that is 46 chromosomes, just get reduced to 23 in number. That is, from diploid number to haploid. Diploid to haploid, or n 2 and 2 n. Okay, anyway, we can say that this meiosis reduces the chromosomal number. It occurs in diploid germ cells to produce. It occurs in diploid germ cells to produce haploid gametes. To produce haploid gametes. Okay, it leads to the haploid phase in the life cycle of sexually reproducing organism. Fertilization restores the diploid phase. That is, we said that after meiosis, the gametes formed are haploid. So how then it is restored? Diploid number 2 and number is restored because during fertilization, both male and female gamete, both which 23 chromosome, not 23 pairs, 23 chromosomes fuse together to form the normal to retain the normal 46 number of chromosome. This is one of the event that occurs in during fertilization with regard to meiosis. Clear with that? Next, we are going to say about the stages of meiosis. Meiosis consists of two stages, that is meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. Meiosis 1 is further divided into two stages, that is a karyokinesis and a cytokinesis. In the same way, meiosis 2 has also two stages, karyokinesis and cytokinesis. The karyokinesis of meiosis 1 is further divided into four phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, and this prophase, anaphase, prophase, metaphase, anaphase, and telophase 1. Okay, so this prophase is further divided into five substages. Okay, That is the most important topic of this chapter. Leptotene, zygotene, pachytene, diplotene, and diakinesis. Okay, leptotene, zygotene, pachytene, diplotene, and diakinesis. These are the five stages of prophase one of karyokinesis, and it has another cytokinesis one. Okay, in case of meiosis 2, it has karyokinesis 2 and cytokinesis 2. Karyokinesis 2 further having five stages: prophase 2, metaphase 2, anaphase 2, telophase 2, and that stage 2, meiosis 2, is known as the equational division. That is, it is almost equal to that of mitosis. First division is reduction division, that is meiosis itself. Second division similar to mitosis, reduces the diploid nucleus to form the haploid nuclei, produces to form haploid nuclei. This produces meiosis 2 produces 4 haploid cells. The only difference is that meiosis 2 occurs in haploid cells and mitosis occurs in diploid cells. All others are equal. Clear with that? So let's move on with meiosis. It is the division of diploid germ cells that reduces the chromosomal number. Once again, let me change the color. That reduces the chromosome number by half the for halving by half forming haploid daughter cells. That is what we have discussed. The 23 pairs of chromosome, 23 pairs. That is what the 46 chromosomes. The 46 chromosomes get reduced to the 46 chromosomes get reduced to 23 in number. So it occurs during the process of gametogenesis. Gametogenesis, what the formation of gametes, that is, it occurs in haploid germ cells. That is gametes producing cells. It occurs in it occurs in diploid germ cells. One second, one confusion. Yes, I just searched whether it was diploid given there or not. Okay, so it occurs in diploid germ cells to produce haploid gametes. Clear with that? It leads to haploid phase in the life cycle of sexually reproductive organism. Fertilization restores the diploid phase. Oh, this is what we have learned there. Okay. Then, key features of meiosis. Meiosis involves two cycles, that is the meiosis 1 and meiosis 2, but only a single cycle of DNA replication. It involves pairing of homologous chromosomes and recombination between them. All of you know that chromosomes, what are they? This is their structure, right? My god, why it is like this? All of you know the structure of chromosomes, right? Okay, just imagine it's like this. Okay, so combination, that is two chromosomes comes in contact and crossing over occurs there. Okay, that point crossing over occurs. Look, this point, the crossing over occurs. So then what is crossing over? Crossing over is the exchange of genetic material between two parents. Okay, two cells, between two cells. Okay. Hmm. Exchange of genetic materials. Okay, so look, this part is the part where the crossing over occurs, and such a part is known as the chiasmata, the portion of the chromosome where crossing over occurs is known as chiasmata. It involves pairing of homologous chromosomes and recombination between them. Recombination refers to the combination of genes. Meiosis 1 begins after replication of parental chromosomes to form sister chromatids at the S phase. 4 haploid cells are produced at the end of meiosis. Meiosis 2. So at the end of mitosis, how many cells are produced? 2 daughter cells are produced, right, at the end of mitosis. At the end of meiosis 2, 4 daughter cells are produced. Clear with that? So stages of meiosis. Meiosis 1 consists of prophase 1, metaphase 1, Anaphase one and telophase one. Oh my god. And telophase one and meiosis two consist of prophase two, metaphase two, anaphase two, and telophase two. Okay, so this prophase one of meiosis one, prophase one further divides into five stages that we have discussed: leptotene, zygotene, pachytene, diplotin, diakinesis. Okay, these are the five stages of meiosis, prophase one of meiosis one. Clear with that? So, meiosis one, it is typically longer and more complex. So, prophase one, we are going to explain start now. Okay, the prophase one is going to start now. Okay, it is typically longer and more complex, including five phases based on the chromosomal behavior. First is leptotene, zygotene, pachytene, diplotin, and diakinesis. So these are the further five subphases of prophase one. Clear with that? So we are going to learn all these stages in detail. Each of the figures should be there in your mind because it will be asked for the examination. Diagram will be given. So leptotene or leptonema. What happens in this stage of leptotene? The chromosomes form a bouquet arrangement. The chromosomes form a bouquet arrangement. What is bouquet? All of you know about bouquet, right? For invitation, for a welcome, we give a bouquet. For example, in case of marriage, in marriages also we have the terms of bouquet, right? So as a formal invitation, we give this bouquet. So this chromosomes form a bouquet arrangement in case of this leptotene stage. That is, look this. Okay, okay, this chromosomes are really curled. Okay, it's not the problem with the pen, it's curled. Okay, in the leptotene stage, it's what happens: the chromosomes form the bouquet arrangement along with the centrioles. Astal rays moving starts to moving apart, starting to move apart. Okay, the nucleus enlarges. Clear with that? Tonema. So pairing occurs, that is water hormone occurs, and synapse. Then what is the synapse forms? Certain chromosome occurs. So that's great. Here, oh my, six these chromosomes, and that complex, the synaptonymal complex. Clear with that? Okay. So chromosomes become more content, similar chromosomes start pairing together, and that is known as the synapses. What is synapses? Synapses or synapses. It is the pairing of homologous chromosomes. It is known as synapses or synapses. Then what is synapse? It is the junction of two adjacent neurons. Okay, so just keep up with the just not this difference synapse and synapses. Okay, so each pair of homologous chromosomes is known as a bivalent. Each pair of homologous chromosomes is known as a bivalent. Clear with that? Next we have the next stage known as the pachytene or pachynyma. Okay, so comparatively longer phase, bivalent chromosomes split into similar chromatids. This stage is called tetrad. So tetrad formation occurs. That is bivalent, clear and distinct. Bivalent becomes clear and distinct, leading to a tetrad. Look this. This is what is said to be a tetrad. Okay, so appearance of recombination nodules at the cross at which crossing over occurs. Then what is this crossing over? Crossing over, as we have discussed, it is the exchange of genetic material between non-sister chromatids of homologous chromosomes. Exchange of genetic material between non-sister chromatids of homologous chromosomes is what is said to be the crossing over. And the point of crossing over is known as what? Chiasmata. Okay. So fully formed synaptonymal complex. Synaptonymal complex becomes fully formed. Okay, that is becomes that is between two homologous chromosomes, and recombination is completed by the end of pachytene. Recombination gets completed by the end of the stage of pachytene. Next we have diplotin or diplonyma. The main or the hallmark event is the terminalization of chiasmata begins. As the chromosomes becomes distinct, the chiasma or the chiasmata moves to its terminal part. Look, this is what is said to be the chiasmata, right? This point. So that moves to the this since that chromosomes are tightly wound and stretched. What happens? The chiasmata moves to the terminal part. Dissolution of synaptonymal complex begins. Synaptonymal complex starts to get dissoluted. Desynapsis occurs. What is synapsis? Pairing of homologous chromosomes. So desynapsis occurs. What is desynapsis? Desynapsis refers to desynapsis refers to opposite of synapsis, right? Pairing of homologous chromosomes. So non-pairing of homologous chromosomes starts to occur. X-shaped structures called chiasmata are formed. So the chiasmata, as you see, it is X-shaped structures. In oocytes of some vertebrates, diplotin lasts for months or years. In oocytes of certain amphibians, vertebrates, etc., the diplotin stage may last to 3 months to 1 year. 3 months to 1 year. And such a stage is known as the suspended diplotin or dictyotin stage. Such a stage is known as the suspended diplotin or dictyotin. Dictyotin. Okay, clear with that? The next stage we are having is with, and the longest phase in prophase 1, it is the diplotin stage. Diplotin stage is the longest phase in or longest stage in prophase 1. Next is the diakinesis. So diakinesis, this terminalization of chiasmata, it is completed in diplotin stage itself. Okay. So chromosomes are fully condensed. Meiotic spindle fibers originate from the poles to prepare homologous chromosomes for separation. Nucleolus and nuclear envelope disappears. Synaptonymal complex dissolution is completed. Complete dissolution of synaptonymal complex. Chromosome become more condensed, shorts, and chiasmata slips off to the top, and terminalization is clearly visible. Since the chromosome is tightly condensed, the chiasmata slips off to the top, and it is clearly visible in the stage of diakinesis, and spindle fiber formation starts. spindle fiber formation starts. Okay. Next is the that's all for the prophase one of meiosis one. Next is the metaphase one having the from meiosis one. Okay, metaphase one. What happens? Spindle fiber formation is completed there, starting from diakinesis. As the chromosomes align on the equatorial plate, so the chromosome gets aligned on the equatorial plate. Oh my god. Chromosomes get aligned on the equatorial plate. Microtubules from each spindle from the spindle attached to the pair of homologous chromosomes. Microtubules from the spindle attached to the pair of homologous chromosomes. And this stage contains double metaphase plate. Look here, it contains double metaphase plate. Whereas in case of mitosis, it has a single equatorial or metaphase plate. Clear with that? Next is the anaphase one. Next is the anaphase one. What happens in anaphase one is that no splitting of centromere occurs. No splitting of centromere occurs, whereas the bivalent split, the bivalent chromosomes get split. Look here, this gets splitted. This gets splitted up and move towards the poles. Okay, so the homologous chromosomes separate while sister chromatid remain attached to the centromere. So no splitting of centromere occurs, and this is the shortest phase of meiosis one. And phase one is the shortest phase of meiosis one. Okay, and Mendel's law of segregation are clearly seen. Mendel's law of segregation is clearly seen in this stage of anaphase. Okay, next is the telophase. The nuclear membrane and nucleolus reappear and two haploid daughter nuclei are formed. This is called the dyad. So disappearance of spindle fibers. Reappearance of nuclear membrane and nucleolus. Each nuclei consisting of half number of chromosomes, but it consists of a double amount of DNA. It consists of double amount of DNA but half number of chromosomes. That is what happening in this phase one. And after telophase, after telophase, what happens is that uh, uh, followed resting meiosis divisions is interkinesis. The of the from that differences are there in mitosis with force 2. Immediate nucleus opposite poles of force. So this attacks with sides the with the splitting of centromere. With the splitting of centromere. Splitting of centromere occurs in meiosis 2 and a phase of meiosis 2. In telophase 2, the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis following, resulting in the formation of tetrad cells, that is, four haploid daughter cells. So, four haploid daughter cells are formed after cytokinesis of telophase 2. So, these are the overall steps of meiosis, meiosis 1 and meiosis 2. Okay, so before moving on to the significance, we must mark certain differences between the mitosis and the meiosis 2. Okay, certain differences. Okay, so the first point that is the difference is that this is mitosis, this is meiosis 2. Not simple meiosis, this is meiosis 2. Okay, so this mitosis is always followed by a DNA replication. This mitosis is always formed by a DNA replication, but meiosis doesn't have such a condition. Okay, so the daughter cells formed after mitosis is exactly similar. Identical offsprings are formed in case of mitosis, whereas the offspring formed in meiosis 2 may be similar or dissimilar. They may be similar or dissimilar. Mitosis occur in diploid somatic cells. Mitosis occur in diploid somatic cells. Okay, and meiosis occur in haploid germ cells. Meiosis too occur in haploid germ cells. Okay, clear with that? That are the 3 points of difference. Next, we have the significance of mitosis. What are the significance? Crossing over takes place. Crossing over takes place leading to a genetic variation, which is important for evolution. Okay, so increases genetic diversity and evolutionary range, and it conserves the chromosome number of each species by reduction division. That is 23 chromosomes of male gamete gets fused with 23 female chromosomes of female gamete to retain the normal 46 or the diploid number of chromosomes. Clear with that? Next, we have a video slide, small video. So just we are gonna play that. So just wait. We are gonna start it. Have you ever wondered why children do not look exactly like their parents? Or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis in special cells called germ cells within the gonads of males and females. For
Gradual reduction in the number of chromosomes to occur in the
Form the number of two in number and kinetochore. So splitting from centromere doesn't occur, whereas the homologous chromosomes get separated, and this is how the cytokinesis occur. And after which we have an stage of interkinesis.
Since the final product of meiosis one is two haploid cells, meiosis two begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase two, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase two, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase two.
So double metaphase plate are shown. And splitting from centromere occurs in anaphase 2 of meiosis 2.
Ends with telophase two, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the two daughter cells into four haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child is identical to either parent. But why are diversity comes from several sources. prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. Pairs of homologous chromosomes align independently on downs.